The endocannabinoid system plays critical roles in many biological processes, ranging from pain sensitivity to immune function. Everything we know about this elaborate system was discovered within the last 50 years. It all began with a simple question about an ancient medicinal plant. Cannabis is an herbaceous flowering species indigenous to Eastern Asia, with records of therapeutic and recreational use dating back more than 45 centuries. While the clinical effects of cannabis on basic body functions were well known, the chemical constituent responsible for its actions remained a mystery until 1964, when an Israeli chemist named Raphael Mechoulam identified and isolated delta-9 tetrahydrocannabinol, or THC, for the first time. Further exploration of how THC and other phytocannabinoids worked were hindered by the limited technologies available to molecular biologists and chemists at the time. It wasn't until 1988 when THC was found to bind to a receptor known as CB1, which is abundantly expressed in the brains of vertebrates, particularly in regions responsible for cognition, emotions, appetite, and motor coordination. THC's ability to attach to CB1 receptors in the brain explained many of its psychotropic and euphoriant effects, but it couldn't explain its peripheral actions. In 1993, a second cannabinoid receptor subtype, known as CB2, was discovered and later found to occur throughout the immune system, gut, and other peripheral tissues. While scientists were solving old mysteries about cannabis, a new and bigger question was emerging. What physiological purpose do these receptors serve? Nature wouldn't conserve them unless they performed a natural physiological function. Attempts to identify endogenous substances that activate these receptors began and soon culminated in the discovery of endocannabinoids in the mid-1990s. Since these landmark contributions, ample research has established that the role of the endocannabinoid system is to maintain homeostasis, or balance. It ensures that cells communicate effectively, but not excessively. The endocannabinoid system is comprised of three components. First, there are the endocannabinoids that the body makes from lipid precursors. Anandamide and 2-AG are the most well-researched and abundant endocannabinoids the body makes. The second component consists of the receptors that these substances bind to, which include, but are not limited to, CB1 and CB2. The third part consists of enzymes that tightly control the levels of anandamide and 2-AG at any given time. The endocannabinoid system is largely under enzymatic control, and the levels of endocannabinoids are determined by the balance of enzyme-mediated synthesis and degradation. The CB1 receptor predominates in the central nervous system, where it functions to restrain excessive neurotransmission. When neurons release excessive amounts of chemical messengers, endocannabinoids act as a brake to restore balance. Here is a schematic of a neuronal synapse in which an excitatory neurotransmitter is being released from the presynaptic cell. The neurotransmitter binds and activates the receptor on the postsynaptic neuron. This stimulation triggers the synthesis and release of endocannabinoids. These endocannabinoids travel in a retrograde manner back across the synapse, where they bind to the CB1 receptor located in the membrane of the presynaptic cell. Once activated, the CB1 receptor inhibits further release of the stimulating neurotransmitter, eliciting a relaxant effect. The CB2 receptor is distributed throughout peripheral tissues, but is highly enriched in cells belonging to the immune system and GI tract. Endocannabinoids activate CB2 receptors located on the membranes of cells. Once activated, CB2 receptors elicit many immunomodulating effects, which depend on the cell type and its environment. CB2 activation moderates cytokine release, which has broad clinical implications. Endocannabinoids interact with many other molecular targets beyond CB1 and CB2. For example, anandamide binds, activates, and desensitizes an ion channel known as TRPV1. This interaction reduces the perception of pain. Easy ways to support the normal function of the endocannabinoid system include exercise, weight management, stress management, and minimizing environmental toxin exposures. Endocannabinoids are made from membrane phospholipids. Most of them are conjugates of fatty acids and amines. Given the diversity of fatty acids and amines that are already in existence throughout the body, there are many possible combinations. Anandamide and 2-AG are the most well-researched endocannabinoids, but the list is likely to expand in the coming years. Hemp is a complex natural product with hundreds of constituents, including over 100 different phytocannabinoids, most of which still await pharmacological exploration. The most abundant and well-researched non-psychotropic phytocannabinoid is cannabidiol, or CBD. At first glance, CBD is structurally similar to THC, The only difference is the broken ring in CBD. This nuance enables CBD to bend in 3 dimensions, whereas THC is flat. This difference explains why CBD lacks euphoriant or psychotropic properties, because it cannot bind to the same site the way THC does. Instead, it works by interacting with a number of other molecular targets that include receptors, enzymes, and ion channels. CBD increases levels of the endocannabinoid anandamide by inhibiting its reuptake and degradation. As a result, more anandamide is available to activate presynaptic cannabinoid receptors. The enzyme that CBD modulates is known as fatty acid amide hydrolase, or FAAH. This inhibition is relatively weak, however, it is supported by multiple studies. CBD may negatively regulate CB receptor activation in the presence of agonists, such as THC. CBD facilitates neurotransmission mediated by the serotonin 5-HT1A receptor. Activation of this receptor transmits calming signals in certain brain regions. CBD also activates and desensitizes neuronal ion channels involved in sensory transmission. Other molecular targets include adenosine, PPAR-gamma, and another G-protein-coupled receptor called GPR55, which may be a novel endocannabinoid receptor. Cannabis is prolific in generating secondary metabolites called terpenes, which accumulate in the oil. Plants make terpenes mainly to defend themselves by discouraging herbivores and insects from consuming the plant. Terpenes lend unique fragrances and tastes to culinary herbs, spices, and plant foods. Many of them have medicinal properties. Beta-caryophyllene is a sesquiterpenoid that not only occurs in hemp oil, but in the essential oils of cloves, oregano, rosemary, and black pepper. In contrast to CBD, beta-caryophyllene can directly and potently bind and activate the CB2 receptor. Current research is clarifying the translational significance of this selective and direct CB2 agonism as it relates to immunology and other areas of medicine. Beta-caryophyllene may be a key contributor to the overall efficacy of hemp oils, but not all hemp oils contain significant levels or guaranteed potencies. Phytocannabinoids are inherently difficult to absorb due to lipophilic properties imparted mostly by their fatty side chains and ring systems. Terpenes are also fat-soluble and poorly absorbed. The bioavailability of CBD ranges from 6 to 19%, with an erratic pattern of uptake from the GI tract and high inter-individual variability that likely contributes to the differences in patient responses. Lipophilic substances cannot dissolve well in the aqueous environment of the intestinal tract. Colloidal delivery enhancement technologies are designed to improve solubility of lipophilic compounds, improving absorption. When selecting a product, look for a broad-spectrum, THC-free extract with a guaranteed potency of CBD and beta-caryophyllene. Quality considerations include country of origin, extraction method, and whether every batch is tested for solvents, pesticides, and other contaminants. A high-potency, enhanced bioavailability product from a source you can trust can make all the difference in patient outcomes.